Good afternoon, everyone. This is Dr. Deepak Sharma from Nirwan University, Jaipur, and I am a biotechnologist. So today we will discuss a topic that is an introduction to mass multiplication procedure for tissue culture. So basically, tissue culture, very common term in biotechnology and life sciences. Nowadays, it is very famous to multiply the plants according to the need, according to their space. So here we are starting it. So tissue culture is the in vitro aseptic culture of cells, tissues, organs, or whole plant under controlled conditions. Nutritional requirement will be there, environmental conditions will be there. The resultant clones are two type of the selected genotype, means whatever plant we will get, they are genotypically same to their mother plants. The control conditions provide the culture and environmental conditions for growth and multiplication. These conditions include proper supply of nutrient, pH, medium, antibiotics, adequate temperature, and sunlight, humidity, moisture. Everything we have to customize for the adequate plant growth. So basically, if we are talking about the requirement of tissue culture, so first major requirement is your explant. Second requirement is your nutrient media. And third requirement is aseptic environmental condition. So these three are the major requirement for conducting a tissue culture experiment for mass multiplication of the plant. So first is explant, that the plant we are using to multiply. Okay, so this is called explant. This is the important part of our micropropagation experiment. Second is nutritional media. Nutritional medium is out of the soil. We are providing some natural, some artificial environment where we are providing nutrients, antibiotics, vitamins, some gelling agent, some hormones, and carbon source. So these are we are providing in nutritional media. Third is aseptic environmental condition, where we are controlling the environment. We are controlling temperature, moisture, humidity, sunlight, to provide the adequate environment to our plant. So mass multiplication, what do you mean? What we mean by mass multiplication? So it is plant tissue culture technology is being widely used to large scale plant multiplication. So where the seeds are not available, multiplication material is not available, and some material not available which is being used for the production of something, or for the multiplication of the or agriculture commodity, there we are using this procedure. In the case of some specific crops, like some medicinal crops, some herbs, and some other crops like banana and sugarcane, recently we are using this mass multiplication procedure, and we are using the plant tissue culture and micropropagation method that is very capable of multiplying the plant material in a short place in short time. Apart from their use as a tool of research, plant tissue culture is a culture technique have in recent year become very popular and important method for the plant propagation and the plant multiplication. So small pieces of a plant, or we can say tissue, that can be used for the multiplication or for the production of the plant further, that is known as explant, as discussed in previous slide. Some how this plant tissue culture or micropropagation technique for mass multiplication is famous. Why? It is famous for endangered, threatened, and rare species, where it is successfully been grown and conserved by micropropagation because of high coefficient of micropropagation in small amount of numbers of initial plant and space. So these are the positive thing. We require very small space, and we require very limited time and we can multiply plants very frequently and very speedy. So from one plant we can produce millions of plant in a year. It depends upon the area available and the skilled person available. So this plant tissue culture is multi-stage procedure basically. So it start with the selection of superior clones and end to the multiplication of plantlet. So selection of superior clones, this is the very important step where the quality of the end plant depends. So we have to choose the superior clones on the basis of their performance. What are their performance, their history, disease free plants, growth. So these are the parameters for selection of a superior clone. So what we would like to choose, the plant performance should be very good in terms of the production, plant or the genotype history should be very strong, then plant should be disease free and the growth is very good in the adverse condition also. Secondly, first we will choose the clone and then we will identify the explant we have to take in the micropropagation experiment. For example, if we are having a plant, we are taking axillary buds as a explant or some meristematic tissues we can take. So these explant we are extracting and then collection of young branches and shoots. So shoots are more preferable because they are frequently growing and they are free from the viruses and bacteria. So we have to introduce this explant into this medium, this nutritional medium. So nutritional medium in vitro culture, basically we are using MS media. So MS media is a media which is preferable in plant tissue culture experiment. The modification we can do, we can customize this media on the basis of our requirement, like vitamins, like hormones, like carbon source, like agar. We can change everything and we can prepare media according to our need and according to the our plant need. Beside this, B5, Gomberg, N6, niche. These are the common types of media used in plant tissue culture according to the need of the or experiment. If our experiment, for example, if our explant is pollen, then we have to use this niche media because pollens needs more energy. So carbon source, the amount of carbon source will be around 40 to 50%. It is generally in the range of 20 to 30%. Sorry, 2% to 3%. 2% to 3%. But in the case of pollens, they need more energy. So the amount of the carbon source will be 8 to 10%. So these medias basically we are using in the plant tissue culture experiment. Then after, our plant will generate shoots, then roots, then we can use this plant, complete plant with shoot and root. We can use this plant for the plantation or in the or agriculture field. So like in this picture you can see, so this is the from our experiment and procedure will start from this slide. So firstly we are introducing our explant into media in the nutritional media. Okay, so the media may be MS media supplemented with oxygen and cytokinin, maybe sometimes gibberellin. Then this shooting will be there in second stage. After 2 3 weeks shootings will be there, means the explant grow, and the shoots start to multiply. So each shoot will give rise to a new plant. Each shoot will give rise to a new plant. Then rooting will be there. Rooting will be there. For shooting and rooting, we are adding separate hormones. We will discuss in next slide. Then after this hardening, we have to do. This is the most important procedure. Hardening. Hardening is very important. So these can be divided into two parts. So first is primary hardening, secondary hardening. So this is generally called first. So there will be two steps. So after completion of our tissue culture experiment, we will get so many propagules, so many multiplied small plant, but they are very young and new. They cannot adopt in the natural environment. They cannot bear the high sunlight. They cannot bear the pressure of low humidity. So what we have to do, in private, in these plant firstly transferred to the nursery or some polyhouse. Nursery and polyhouse. Nursery and polyhouse. So we are transferring these plants from our lab to nursery or polyhouse. So this is called primary hardening. This is called primary hardening. So then we want our plant to adopt the natural environment. So then we will transfer this plant into field. So this is the secondary hardening. So we have to make our plant to adopt the natural condition, high sunlight and lower humidity. So firstly we will keep our plant in nursery, okay, and then we will transfer our plant into field. This is called secondary hardening. So hardening is very important process, and during this process, after when our plants are ready to shift in field, so primary hardening and secondary hardening, around 10 to 20% plant will die. 10 to 20% plant we will lose during this procedure. So this is the very important procedure. We have to complete this procedure very carefully. So what conditions do plant need? So actually, what are the requirement? The basic requirement is explant, secondly nutritional media, and third is aseptic culture conditions. So one by one we will discuss these requirement into detail. So firstly, a protocol. Each and every plant, each and every plant need a separate standard operating procedure, a standard procedure for the multiplication, mass multiplication of the plant. So if, for example, we are having some different protocol for herbals, we are having different protocols for aromatic plants, and we are having some different protocols for sugar cane and for banana. Protocols are also differ for the according to the requirement of the micropropagation or purpose. Likewise, if we want a experiment of plant tissue culture for the production of secondary metabolites, then we have to use some semi-solid media. We have to use agar in a lower concentration. So when our target is to get shooting first and very fast, then we have to add kinetin, zeatin, 2IP, TDZ, these kind of cytokinin we have to add. If our requirement is rooting, then we have to use oxygen in more concentration. If we are targeting callus, then we have to add both the hormones in equal concentration. So protocol is totally depend on our requirement and our target. Okay, so we will discuss the basic protocol and basic multiplication procedure. So this is the optimum protocol. You can find this protocol in so many research papers are there, and some videos are there, some literature available already on the internet. You can easily find this protocols. Appropriate tissue, that is explant. So explant is any plant part or any group of parts, we can say single cell or maybe tissue, maybe organ, maybe anything from we are getting from the plant, that is being used as a initial plant, initial material. It is known as explant. It is very important. We have to sterilize it. We have to select the elite genotype for this. A suitable growth medium containing energy source as an inorganic salt to supply cell growth needs. This can be liquid or semi-solid. So two type of media will be there. One is solid, second is semi-solid. Both we can use for the production or for the multiplication of the plant or for the another purpose as discussed. So these growth media should contain energy resources. So energy resources will be or carbohydrate. So we are using carbohydrate in this concentration 2 to 4 gram per liter. 2 to 4 gram per liter means 0.2 to 0.4 percentage agar if we are taking weight by volume 0.2 to 0.4%. Then inorganic salts we have to add. Some cell growth needs means auxins and cytokinins. Okay. Third main thing is aseptic conditions. Aseptic sterile conditions we have to provide. These microorganisms will grow faster than plant or animal cell. Bacteria and fungus will grow faster than animal and plant cell. So we have to be very careful during conducting the experiment and we have to avoid the contamination. For this we can use some fungicides, from some antibiotics we can use. We will discuss in the next slide. Growth regulators, both auxin and cytokinins we will use as discussed. Frequent subculturing to ensure adequate nutrition and avoid the buildup of waste metabolites. So when we are taking explant and we are cutting it into small pieces to start the experiment, what happened? It start to leaching out some polyphenols. So when they come in contact with oxide, so they produce secondary metabolites. So what they will do? They will start to make the media brown. So this is called browning. Browning. So that's why frequent subculturing is required to avoid the browning. Browning can be also avoided by using the ascorbic acid, citric acid, polyvinyl pyridine, diethyl ascorbic acid. So these can be used to remove browning also. But this is very harmful for our plant because these metabolites produced after browning will inhibit the activity of enzymes ultimately. So we have to avoid this browning. So first basic need is appropriate tissue, that is called explant. It's cell, tissue, organs of a plant that is used to start in vitro culture. So we are starting our culture by using this plant. Many different explant can be used for tissue culture, but axillary bud, that is important. This axillary bud is required, then meristematic tissues are required. Why we are taking these type of tissues? Because they are very highly growing. So due to their high growth, virus and bacteria cannot multiply in these explants. So that's why we are using these explants. So the explant must be sterilized. So explant we are using, it is already explored to the outer environment. So it is usually it is having some bacteria or fungus, some dust and some other type of impurities on their surface. So what we have to do? We have to chemically sterilize it. We have to chemically surface sterilize it. Surface sterilization. Surface sterilization we have to do. So we are having a different type of material in this tissue culture experiment. Like for explant, we have surface sterilization method. For some vulnerable chemical material like antibiotic and hormones, we are having some another type of sterilization, sieve sterilization, filter sterilization. And for laminar flow, we are having some radiation sterilization, we are using UV rays. Okay. And media sterilization, we are using autoclave. So different kind of sterilization process we are using in this experiment according to the material we are using. So the explant must be sterilized to remove microbial contamination. This is usually done by chemical surface sterilization, where we are using HgCl2, NaOCl3, alcohol. So these kind of chemicals we are using to remove the impurities and bacteria, fungus from the upper side and surface of the plant. So these are the agents such as bleach in different concentration we are using to remove the pathogens. After tissue injury, dissections such as compound will be oxidized by polyphenol oxidase. Tissue culture turn brown. So this is called browning. So whenever we are dissecting the tissue for the procurement of explant, some polyphenols oxidized by the some polyphenols oxidize. So that produce some material that make tissue turn brown or black. So for this purpose, what we have to do, we have to use this method for the overcoming of the browning. We can use the chemicals such as citric acid, ascorbic acid, polyvinyl pyrrolidine, and diethyl activated charcoal, and pre-soaking explant in antioxidant can also use to overcome this problem. So these chemicals we can use. So basically the major chemical we are using, it is ascorbic acid, and this activated charcoal commonly we are using in tissue culture media. This is the second method, pre-soaking explant in the antioxidant. So it can uh, destroy the free radicals and oxidase, so that browning can be overcome. Third method is incubating the initial period of the culture in a low light, reduce lights or darkness. So initial culture we can use the low light for the removal or for the avoiding of the to avoid the browning of the media. Another method is frequent transfer into fresh medium. Frequent transfer into fresh medium. Sometime subculturing can be subculturing we are doing due to two reasons. So first is browning, and second is media change. So first thing we have discussed, we already discussed about the browning, what are the method for overcoming the browning. Okay. And second, why we are doing subculturing for media change. So in long protocol and long experiment, the media start to deplete. All nutrient will start to deplete and media become dry. So it will be very harmful to our growing plant. So that's why we have to change the media. And in the case of browning also, in 80% cases of browning, we are changing the media because it becomes very brown, and these metabolites start to inhibit the enzymatic activity. That's why we have to change it. Nutritional media, this is also an important part. Nutritional media, basically media we are using, it is very common media we are using, MS media with the different concentration of enzymes, antibiotics, other additives, carbon source. These can be altered or these can be customized. So MS media given by Morashige and Skoog. These two scientists formulated this media. This media is in the 95% cases in tissue culture experiment we are using MS media with altered concentration of the other components. Okay. So other media, B5 media, N6 niche media, Gombak media, but these media can be used according to the requirement. So when an explant is isolated, it is not no longer able to receive nutrient or hormone from the plant. So we have to transfer these plants into the media frequently. In addition to the culture must be provided with the ability to extract the waste product of cell metabolism. This is accomplished by the culturing on or in a defined culture medium which is periodically replenished. So media should be chemically defined. What kind of nutrients needed to the explant and what is our purpose? In nutrient media is defined by mineral salt. So these are the important mineral salt first. Second is carbon source. Third is vitamins, plant growth regulators, and other organic supplements. So these are the part or these are the component of a nutritional media. So all are having some different functions in the media. pH determined. pH is also very important. It should be 5 to 6. pH range should be 5 to 6, and during autoclave it will go down. So that's why we have to set the pH already. If we want to set pH 5.5 final pH, then we have to set it to 6, because after the autoclave process the pH will lower down by 0.5 to 0.3 to 0.5. Now one by one we will discuss all the components. Like first is mineral salt. So when the plant is growing in natural conditions, they are getting uh, micro and macro elements from the soil. So that's why we have to provide all the macro and micro elements like sodium, potassium, calcium, magnesium, sulfur, phosphate, magnesium, zinc, boron, cobalt, copper. These nutrients we have to provide, and for this we are adding this different mineral salts available. So macro element and micro element. So two type of elements we can divide, which are needed into plant tissue culture experiment. So first is macro element. What is the definition of macro element basically? So macro elements are those elements which are required in the concentration of greater than 0.5 millimole per liter. The elements which are required in concentration of more than 0.5 millimole per liter, then they are macro elements. So for example, nitrogen, potash, potassium, calcium, sulfur, magnesium, chlorine. So these are the macro element because they are needed in good concentration. Secondly, micro elements, the elements which are required in less than 0.5 millimole per liter. Okay, so these elements are ferrous, manganese, boron, cobalt, copper, zinc, molybdenum, iodine. Okay, and maximum growth rate means the optimum concentration of these elements. If we are using the optimum concentration of these elements, we will get the maximum growth rate of our plant. Next component of our media is sucrose or glucose, that are the carbon source. Energy source of our explant is sucrose and glucose. Sometimes fructose we are also using. 20 to 40, it means 20 to 40 gram per liter we are using according to requirement. As discussed, in the case of pollens we need some more energy, so we are using to 6 to 8 or 10 gram per liter sometime, but in common experiment we need 20 to 40 gram per liter is sucrose and glucose. Absolute requirement B1. If we are talking about the vitamins, B1 is also required. B6 paradoxin is required. Beside them, pantothenic acid, B5, folic acid, biotin, p-aminobenzoic acid. These are the required in addition to the ascorbic acid, that is vitamin C. So these are the vitamins and carbon source requirement. Plant growth regulator. These are the this is the very important part, because these hormones regulate or control the redifferentiation of the plant. Okay, so So first auxin, second is cytokinin, third is gibberellin, and in some time we are also using abscisic acid. In the case of embryogenesis, induction of embryogenesis, we are using abscisic acid. So first, if very most important, auxin. So what are the major work of auxin? It is cell elongation, cell division, callus formation, and swelling of tissue, and the formation of adventitious root. Okay, and it inhibit the adventitious shoot formation and axillary shoot formation. It also inhibit this adventitious and axillary shoot formation. Means auxin maximally promote the root formation. Okay, and these are the major work. If a question comes into examination of plant tissue culture, what are the major work of the auxin, and the options are available: root formation, then root formation is the right answer. So for example, for tissue culture purpose, some specific kind of auxins we are using, that is 2,4-dichlorophenolic acid, naphthalic acetic acid, indole acetic acid, and indole butyric acid. So different concentration of these kind of auxins we are using according to the demand. Okay, so generally we are using the concentration, it is range from 300 to 500 ppm. This parts per million concentration we are using for the hormones, because they are needed in very low quantity. Cytokinins are the second class of the plant growth hormones which are needed. So cytokinins, they induce or they promote shoot induction and cell division. So it is the opposite. So where auxins promotes the root, cytokinins promote the shoot, and cell division in another work. For example, we are using, basically concentration will be same. We are using benzyl aminopurine, G18, kinetin, thiodiazurone, 2-isopentyl adenine. So these are the examples of majorly using cytokinins in the experiment of the plant tissue culture. Beside this, GA3, that is gibberellic acid. Gibberellic acid, sometimes in some experiment we are also using gibberellic acid. So the work of gibberellic acid is plant regeneration and elongation of internodes. In some cases, like induction of induction of embryogenesis, we are using abscisic acid also. Other media component, in other component, firstly media formulations. Some media formulations are available which are ready to use. We just have to add them in a MS media, and they will help to they will help in growth and development of the plant. So like media formation, we can say in some cases we are using coconut water. Coconut water is also media formulation. So it is already a media formulation, we can say, having so many nutrients and good things for the reproduction of the plant. Natural complexes like charcoal and undefined things we are using, that concentration is not defined, but they are known for the growth and development of the plant. That's why we are using gelling agent agar. Agar is the universal gelling agent. We are using this agar into 6 to 8% concentration. 6 to 8% concentration we are using this agar. Charcoal, we are using organic acids, antibiotic for the removal of the microorganisms. We are using this amino acids and organic salts. We are using to strengthen the nutritional background of the media. That's why we are using. So these are the all media component we are using into MS media. MS media, it is a, so we are preparing. Basically, we are preparing this MS media by combining these all the components. Okay, so the concentration is already defined into some so many literatures. So we have to take these elements, micro elements, macro elements. Secondly, we have to use carbon sources. Then we have to use vitamins. Then we have to use hormones. Then we have to use these other components. Likewise, or otherwise the ready to use media, MS media is available into the market. So all the components are there, just we have to set the pH, we have to add agar, and we have to add only vitamins and hormones, and other things are already added. It is available in 100-gram packing. If we want to use the ready-made MS media, so the concentration will be 4.4 gram. We have to take from this mixture. We have to take 4.4 gram per liter, and then we have to set the pH. We have to add agar, and we have to add only hormones. Then your media is ready. Because it is very complicated procedure, we are adding so many things and so many components. So there will be so many errors. The chances will be high for error. So as we say that we are taking a small plant, we are taking cell or group of cells or organ, okay, and tissue. But how this come possible to generate a whole plant from these small things? Due to one thing that is known as cellular totipotency. Cellular totipotency can be defined as the capacity of a plant to regenerate, the capacity of a single cell, the capacity of single cell or group of cells to give rise to an entire organism. Means a single cell we can use, it will give rise to a whole plant. So this capacity is known as totipotency, and this is the property of only plants. Plants only exhibit this property known as totipotency. Okay, so cultured tissue contain competent cell capable of regaining competence. Okay, so in 1957, Skoog and Miller demonstrate that two hormone affect the explant differentiation. So whenever we introduce the explant, it start shooting, it start rooting, okay, and differentiation start, means callus formation will be there and different parts of the plant will be there. So this process differentiation will be there, but by customizing these hormone requirement, what we can do, we can customize the differentiation according to the need. For example, auxin auxin stimulates root development as discussed, and cytokinin stimulates shoot development. So if we want rooting first, then we can use the higher concentration of auxin. If we will use higher concentration of auxin, root development will be there first, only root builder. And if we are using the higher concentration of cytokinin, then shoot development will be there. If we want to develop shoot and root parallelly, then we have to use the equal concentration or the same concentration of both the component for the callus development. So generally third combination we are using in general. If we want some research and we are conducting some research trials, then we can use this both conditions, auxin high and cytokinin high, according to the need. Clonal propagation, micropropagation. So basically we have discussed the major requirement of plant tissue culture or any experiment for the mass multiplication. So now a term that we are using, that is micropropagation. So micropropagation is a term. Micro means a small part we are taking and we are propagating in a high number. By taking a small part, we are propagating it to maximum. So the art and science of multiplying plant in vitro means in lab condition we are multiplying plant, and we can multiply 1 to 2. We will get from 1 plant to 10 plant in 6 to 8 weeks. Then after we will get 100 plants from these 10 plants. then 1000 plant, 10,000, 1 lakh. So we can multiply plant into the millions according to the area available and the uh, size available and the skill person available. We can multiply plant. Within a year, we can produce millions of the plant. So the commercial production of the plant through micropropagation technique has several advantages. So it is having some advantages over the grafting, cutting, and seed air layering. The success of many in vitro sections and genetic manipulation techniques is higher. Plant depend on the success of in vitro plant regeneration. A large number of plant dates can be produced. Cloned plant can be produced in a small space in a small time by using the single individual. So this is into micropropagation. So it is a vegetative reproduction. Basically, it is asexual reproduction since we are using some somatic part. Some somatic part we are using, and we are producing the clones of the plant. So it is comes under the vegetable or asexual reproduction, where we can produce the millions of the plant. So this is the basic procedure, and some other diagrams we can also find on the website or internet. So it start with the mother plant. As we discussed, we have to choose a elite good genotype from where we can extract or take the explant. So this is our major mother plant. It should be very healthy. It should be disease free. From where we are choosing a plant part that is suitable for taking explant, like meristematic tissues, like axillary buds we can take. So then we are starting our experiment by introduction of this explant into media. So this step is called initiation. Then multiplication will be done. More shoots and propagules will give rise after 4 to 6 weeks. Then rooting will be there. Multiplication, rooting, shooting will be there after this primary hardening. Primary hardening means we are transferring it into nursery or polyhouse, where they will adopt the environment and they will make them strong enough to bear the strong sunlight and low humidity. Then after 3 to 4 weeks, when we assure that the plant will survive in the open environment, natural environment, we will transfer them into field. This is known as secondary hardening. So this is a common protocol, and this is a rotational process. After this secondary hardening, we can take this as a mother plant, and we will take some explant from this plant, and we will again continue in this cycle, and we will produce more and more plant in a year. So as discussed, micropropagation is multi-stage process. So some steps are there, like we can name them 1, 2, 3, 4, 5, 6, or we can name them 0, 1, 2, 3, 4, 5. As available in literature, it start with 0 stage. So micropropagation start with the selection of the plant tissue explant, as discussed. So first explant we have to discuss. Uh, we have to choose the explant. This explant is very important. So leaf, apical meristem, buds, root, anything we can choose, but the more preferable explant will be meristematic tissues and apical buds. So first step is preparation of donor plant. How we will prepare this donor plant, and how we can cut the explant from this mother plant. What we have to do? We have to do surface sterilization. As discussed, plant is having so many debris, dust, microbes, so we have to remove them by using the chemical surface sterilization. And this we are using sodium hypochlorite, mercury chloride, and then alcohol. So subsequent washing with these material will remove all the debris, dust, and microbes. This is the first step. Second step is Initiation stage. Okay. So in first stage we will take the plant only, and in first stage we will do surface sterilization as discussed. We will use this sodium hypochlorite, this sodium hypochlorite, calcium hypochlorite, ethanol, or HgCl2, mercury chloride. They are different condition concentration. Generally we are taking 0.1 to 0.5% concentration of these chemical, because they are very harmful and they are carcinogenic. So while handling these chemicals we have to be very careful. Third stage is multiplication stage. So once we introduce or explant into media, it start to take nutrients from the media, and it start to grow. So we have introduced only one explant, then it start to multiply into propagules, different propagules, like 8 to 10 propagules in a one time will generate from the single explant. Then rooting stage will be there if we have added the auxins as required for rooting, like 2,4-D or IBA, IIA of naphthalic acid. The rooting stage may occur simultaneously in the same culture media. So rooting may be occur in the same media we have started in the protocol, but sometime what happened, due to browning we have to change the media, and we have to transfer this plant into fresh media to introduce the rooting. After that, acclimatization stage, as discussed, acclimatization means the hardening process, primary hardening and secondary hardening. At this stage the in vitro plants and are weaned and hardened. We are doing hardening of the small and new young plants. Hardening is done gradually from the high to low humidity. High to low humidity means in lab we are controlling the condition, we are maintaining the humidity around 60 to 80% and temperature around 25%, 25 degrees centigrade. But in the natural condition the temperature will be around 35 to 45, and the humidity will be around 40-30%. So we have to transfer the plants in a condition of low humidity to make plants some more adaptive to the outer environment, and low humidity, and from low light intensity to high intensity means sunlight will be there and low humidity will be there. So we have to ready our plant. The plants are then transferred to appropriate substrate, sand, peat or compost, whatever we are using, or polyhouse, gradually hardened under the greenhouse. So by using these sand, peat and compost, we can use this plant can be transferred into the polyhouse. So we have discussed the requirement for the micropropagation and plant tissue culture experiment, and the importance of this plant tissue culture experiment, and components of plant tissue culture also, requirement is also. Now why we are using this technology instead of seeds, instead of air layering, grafting, layering, different kind of plant multiplication techniques we are having, traditional methods are having, but why we are using this technological advance or micropropagation? So these are the applications. Screening of cells rather than plants are for advantageous characters, example herbicide resistance. So these studies we can do easily in the lab conditions. We will do the herbicide and resistance, herbicide and bactericide or any other thing, tolerance or resistance studies we can do in the lab conditions. Large scale growth of plant cells in liquid culture. In liquid culture we can grow a large scale of the plant to cross distantly related species by protoplast fusion. So this method, this micropropagation, allow us to produce some hybrids, new hybrid, by crossing two differently, two distantly species, related species, means one is barley and one is wheat, or we can say one is chili or one is tomato. So we are crossing it and we are producing something new, some hybrids we are producing by fusing the protoplast of the both these species. So this can be also possible in this method. But embryo rescue we can do. In some conditions, we cannot save the embryo, like in endangered species, the embryo will destroy whenever we are going to store it. But in the case of plant tissue culture, we can maintain plant and we can use this as a embryo. For the production of double monohaploid plant from the haploid plant, so we can use also double haploid. By using haploid and haploid, we can use double haploid plant as a tissue for transformation, followed by the either short-term testing of genetic construction or regeneration of transgenic plant. So whenever we are in need to check or to uh, to construct some genetic makeup of a plant, or just we have to check the genetic makeup of a plant, then in short term we can produce the plant without depending on the environment, and we can do the genetic studies. Regeneration of transgenic plant also. So transgenic plant we can produce by this method. So whatever method we will use, like indirect method or direct method for transgenic plant production, so many methods are there. Implant is there, agrobacterium based method is there. So all method need plant tissue culture based protocol. We have to conduct the experiment into plant tissue culture lab. Next is in vitro conservation of germ plants. So in vitro, in lab we can conserve the germ plants, like seed we are conserving in our lab, like plant material we are conserving, like corn we are conserving. So as like we can conserve the germ plasm in the tissue culture lab. Advantages of micropropagation. So these the previous slide we discussed the application. Now these are the advantages why we are using this method over traditional method. So first is rapid multiplication of genetically uniform plant. So this is the most important. So first method is, if we want to produce the millions of the plant producing material in a nursery or in a traditional method, it will take so many labors, skilled labor, and so much space also, and so many days. Instead of this, if when we are using plant tissue culture, we can multiply rapidly plant which are uniformly same, genetically same, in a less time, less area. The production of multiple plant in the absence of seeds or necessary pollinators. So we also not in need of the pollinators or any other things which facilitate or cross of the two different species or two different male or female plant. So we just need a only explant. We need not any pollinators or any other things. That is the regeneration of whole plant from a plant cell that has been genetically modified. So we can also produce genetically modified plants by this method. The production of plant in sterile containers that allow them to move in genetically reduced chances of transmitting disease. So since we are using virus free or disease free explants, so we can produce disease free, pest free or pathogen free plants that will be very easy to transfer and move, and there will be very reduced chance of transmitting disease from the one generation to another generation. In traditional agriculture, when we are using some explant or some planting material with diseases or virus, viral diseases, bacterial diseases, seed-borne diseases, they will transfer to the next generation. But in this case, the disease or pest or they will not transfer into the next generation. The production of plant from seed that otherwise have very low chances of germination and growing. In the case of, for example, in the case of orchids and nepenthes, there is very low chances of germination of the seed. So that's why in this case, this condition or this method is very good. The production of plant throughout the year. So this is also a very important benefit of using the plant tissue culture or a micropropagation method, that we can get plant year around. Year around means we are using aseptic condition, control conditions. So we are managing the humidity, we are managing the temperature, we are managing the sunlight. So that's why we can grow plant uh, year around, 12 month year around. In traditional agriculture, we are just dependent on the environmental conditions. We can only grow wheat, mustard, and wheat, mustard, and Caesar arietinum into the Rabi crop, and pearl millet can be only grown during the Kharif crop. So different environmental conditions and climatic conditions are needed. In the case of this technology, these environmental conditions can be overcome, or they cannot be needed. So this is the a protocol or a setup of a lab, or we can say, first start with plant material, surface lation, we are cutting the explant, then surface lation, then we are transferring it into media, then calyx production, then shooting, rooting, and the hardening and growing soil, then plant clones we can produce. So this already been discussed in the earlier slides. So this is a flow chart for the understanding. If you want, these are the reference taken from the web references, and some books are there, some research papers are there, so you can follow these references also. So, thank you so much for your patience and listening of this lecture. Thank you so much.